People are trying them, the media is tooting them, but scientists say we need to know more about how they work and when they can help. This quote comes from the March Stanford Magazine edition, titled "Falling for Psychedelics." So is there hope, or is it all hype? Well, in this video, I'll give an overview to classic psychedelics with some essential neuroanatomy, and then we'll look at this recent study that has identified a possible therapeutic mechanism that involves activation of pathways involved with dendritic growth. As we'll come to conclude, studies like these are imperative for providing a foundation with which to guide optimal dosing and to identify individuals who could benefit from psychedelics, and also the other challenges ahead. So firstly, what are psychedelics and why are people excited about them? Psychedelics are molecules that can affect perception, cognition, and mood, and produce intense subjective experiences. Due to these effects, there is much interest in understanding the therapeutic potential for treating neuropsychiatric disorders such as anxiety, depression, and substance abuse. And this has even been employed in medically supervised psychedelic-assisted therapy. In the magazine article, it describes the story of a former Special Operations soldier and FBI agent, Paul, who, after years of nightmares, insomnia, anxiety, and depression from recounting his time in Iraq and Afghanistan, participated in a trial where he took psilocybin-assisted therapy. After taking the substance, Paul describes his experience as exhausting. It was like he'd been wrestling with himself for 6 hours straight, but afterwards, there was a load off his back, of punishment, guilt, and self-hatred. He goes on to say, "It was not a fun experience, but after, I felt so good. I felt free, hopeful, happy." It is thought that a psychedelic-induced trip might help the brain reorganize thought patterns to find new ways of processing anxiety, depression, or trauma, says Laura Hack, an assistant professor of psychiatry and behavioural sciences who treats veterans of PTSD and depression. And a few other pilot studies have been conducted. Here reported in the New England Journal of Medicine, a phase 2 trial showed that participants with treatment-resistant depression, when given psilocybin at a single dose of 25 milligrams, but not 10 milligrams, reduced depression scores significantly more than a 1 milligram dose over a period of 3 weeks. But it's worth mentioning that 77% of all participants in the study had adverse effects ranging from headaches, nausea, and dizziness. Psilocybin, what Paul took, can be found in more than 100 mushroom species. Along with LSD, DMT, and DOI, these are referred to as the classic psychedelics. One action of these classic psychedelics is that they combine to a protein receptor known as the 5-hydroxytryptamine 2A receptor, or 5-HT2TAR for short. If you take a look at the chemical structure of these classic psychedelics, you'll notice that they resemble the compound serotonin, otherwise known as 5-hydroxytryptamine. Hence, the receptor I just described is one type of serotonin receptor. And I say one type because this is just one out of at least 14 different serotonin receptors. And in the central nervous system, serotonin receptors are found in different regions of the brain, including the cerebral cortex, the outside of the brain, the hypothalamus, the hippocampus, and the brainstem. And this image here shows where the serotonin 2A receptors are localized. And so the 3 chemicals that we'll focus on for the remainder of this video are 2 psychedelics, psilocin, the one derived from mushrooms, psilocybin, DMT, the main psychoactive component of ayahuasca, a potent hallucinogenic plant-based brew that has been consumed for thousands of years by indigenous people of South America, and good old serotonin. And so if we focus on these 3 compounds, and I mean really focus on them, it raises an interesting question. These molecules are mimicking serotonin, and we all have serotonin in our bodies. So why are we not all continuously in a hallucinogenic state? Clearly, somehow, these slight molecular differences are having an impact. But what and how? Well, that brings us on to this recent publication, Psychedelics Promote Neuroplasticity Through the Activation of Intracellular 5-HT2A Receptors. So we come back to the serotonin 2A receptor, which falls under the class of proteins known as the GPCRs, or G-protein coupled receptors. These are, in my personal opinion, a top-tier family of proteins. that have far-ranging functions but are united by the fact that they are embedded in membranes, and they have 7 transmembrane domains, so that they have parts sticking out on both sides of the membrane. This makes GPCRs, like the serotonin 2A receptor, good signal transducers. As many GPCRs are predominantly found at the cell surface, they can transduce a signal from outside the cell inside. So these 3 compounds, you may think that potentially due to the similarity, psilocin and DMT are antagonists. Maybe they're blocking serotonin from binding the receptor. But that's not the case. Like serotonin, these compounds are also agonists. They cause activation. But the impact of these chemical differences altered the chemical's lipophilicity, how much it likes lipids, or another way of thinking about it, how easily these chemicals can pass through the cell membrane without a protein. Serotonin is highly polar, and so cannot easily pass through the cell membrane, but both psilocin and DMT possess extra methylation that makes them prefer lipids, and so they can pass easily into the cell. And interestingly, in the study, what they did was they plotted the lipophilicity against the psychoplastogenicity, and they could see a positive correlation. Here, psychoplastogenicity is defined as the ability to promote structural plasticity in the dendrite branching. So this is perhaps the point of the video where I should do a little bit of a Neuro 101, as neurons receive chemical signals from neurotransmitters at so-called dendrites, and what they are observing here is increased branching and spikes on these dendrites following treatments such as with DMT, as you can see in this figure here. And so what I'm trying to say is what they found in this study is that the psychoplastogenicity correlated better with the lipophilicity of the chemicals than the ability of the chemicals to just activate the serotonin 2A receptor. But how and why? Why would going across the membrane impact this downstream effect of altering the dendrite growth? Well, the researchers decided to look at where are the location of these 2A receptors within the cell. And it turns out in the neurons, in the cortical neurons, they're not just at the cell surface. In fact, they observed bright staining within the cortical neurons. And they showed that it co-localized with other markers within the cell, suggesting that maybe these serotonin 2A receptors are also functioning within the cell as opposed to just at the cell surface membrane. And so to test this a bit further, what they did was they blocked or enhanced the membrane passing ability and saw how it influenced the dendrite formation. My favourite part of the study was when they exploited the fact that serotonin can't pass through the membrane, and instead genetically altered the cells such that they express a serotonin transporter so that the cells could now take in serotonin. And so when they expressed the serotonin transporter protein in the neurons and treated with serotonin, it matched the response that they would see with DMT. So as we can see, it's all about location, location, location. The paper suggests that the therapeutically relevant mechanism of action for DMT and psilocin involves the 2A receptor, which intracellularly promotes dendritic growth and increases the spine density of rat cortical neurons. So I should point out here as well that these studies are done in rat neurons. And so to go back to my first question, is this hope or hype? Well, me being the optimist, I'd like to say there is hope, but there are clearly many areas still to address. Hence why papers like this recent one are intriguing, because they can give an insight into the molecular mechanism, as once you know the target and how it's impacting the cell, you can design or look for more selective chemicals or chemicals that have different properties and use it to guide optimal dosing and identifying individuals who are likely to respond positively to psychedelics. So it's also worth pointing out here that in this paper they primarily considers the serotonin 2A receptor, but another important caveat to consider is that many, if not all psychedelics, also potently activate the closely related serotonin 2C receptor. And then it's also worth thinking about the long-term consequences, both in terms of understanding long-term molecular mechanisms, but I meant more long-term in thinking of standard clinical application. Even when telling people I was making this video, I got some looks when I mentioned the phrase psychedelics. So a big one is public perception. And this interesting commentary suggests changing the name, but for multiple reasons. Narrowly defining a drug class by one set of its effects, psychedelic, colours the perception as it being the only thing that it does. An example they give is antidepressants. if someone told me to take one for a non-psychiatric condition, I might have preconceptions about it because of the class name. And so this commentary suggests alternatives to psychedelics as psychoplastogens or neuroplastogens. Anyway, besides that, there is also the challenge of actually conducting clinical trials, partly because it's hard to control for them. That being, there might be a strong placebo effect, and in the New England Journal of Medicine article I gave earlier, the control condition was just having a lower dose of the psychedelic compound. So clearly there is some reason to be excited about psychedelic compounds for treating different disorders, but I like the comment from this magazine article that today's psychedelic researchers hope the public will wait a little longer for science to provide answers before something bad happens and the promise of psychedelic therapy slips once again back underground. The hype around this partly is because psychedelics could be a quick fix. There are other ways of reducing the hyperconnectivity of brain regions over time, including mindfulness, meditation, exercise, and antidepressants. But what's unique to psychedelics is how rapidly such a dramatic change can happen. But it may not be as long as you think, as there is a lot of research going on at the moment and a lot of momentum, as in the process of making this video, another paper came out showing neuroimaging following human consumption of DMT. And what they saw in this paper was that DMT breaks down the basic networks of the brain, causing them to become less distinct from each other. We also see the major rhythms of the brain that serve a largely inhibitory constraining function break down, and in concert, brain activity becomes more entropic or information-rich. But the other thing that I find quite curious are what are the long-term responses and changes at the molecular level. This study suggests epigenetic changes, which, as I explained in this video, are heritable marks. So with that, I hope you've learned something in this video. Thank you to all my supporters on Patreon, and thank you for listening.